Hello everyone, now we'll be discussing pyrimidine synthetic pathway. Now the source of various atoms which is utilized in the pyrimidine molecule. First one is aspartate, which contributes to 3 carbon atom and nitrogen atom, and glutamine is another source of second nitrogen, whereas the carbon atoms comes from the carbon dioxide. Cut it. Now let us look at the source of various compounds which is present in pyrimidine ring. Nitrogen atom comes from the glutamine amino acid. The carbon comes from the carbon dioxide. The rest of the carbon and nitrogen comes from the aspartate molecule. Now, similar to purine synthesis, pyrimidine synthesis also has de novo and salvage pathway for the synthesis. Now, let us look at this diagram for the synthetic pathway of pyrimidine. Pyrimidine synthesis. So, the carbon dioxide and glutamine in the presence of enzyme carbamyl phosphate synthase 2 forms carbamyl phosphate. Carbamyl phosphate 2 is present in cytosol. There is another CPS1 enzyme which is involved in urea biosynthesis. So CPS1 and CPS2 is present in the human body. So 2 is utilized in pyrimidine biosynthesis. CPS1 is involved in urea cycle. It is a mitochondrial enzyme, whereas CPS2 is cytosolic enzyme. Remember, this is an MCQ both in NEET PG as well as university examination. So this CPS2 is the regulatory enzyme in pyrimidine synthetic pathway. Now the formation of carbamyl phosphate, it gets converted to carbamyl aspartate in the presence of enzyme aspartate transcarbamylase utilizing one molecule of aspartate. This carbamyl aspartate in the presence of enzyme dihydroorotate forms dihydroorotate. Dihydroorotate gets converted to orotate using enzyme dihydroorotate dehydrogenase converting NAD to NADH. These three enzymes, aspartate transcarbamylase, dihydroorotate and dihydroorotate dehydrogenase is an example of multifunctional enzyme. Orotate gets converted to orotidine monophosphate utilizing enzyme orotate phosphoribosyltransferase transferase utilizing one molecule of PRPP. This orotidine monophosphate, written as OMP, in the presence of enzyme OMP decarboxylase leads to formation of uridine monophosphate, which gets converted to uridine diphosphate in the presence of kinase. The enzyme orotate phosphoribosyltransferase transferase and OMP decarboxylase enzyme is an example of bifunctional enzyme. Now the UDP which is formed is acted by kinase leading to formation of UTP. This UTP gets converted to CTP in the presence of CTP synthetase. The UDP which is formed gets converted to deoxyuridine diphosphate in the presence of ribonucleotide reductase utilizing thioredoxin, and this deoxyuridine diphosphate gets converted to DUTP in the presence of nucleoside diphosphate kinase. This further gets activated by DUTPase to form DUMP, which gets converted to DTMP in the presence of thymidylate synthetase, converting N5N10 tetrahydrofolate to hydrogen folate. And this enzyme thymidylate synthetase is inhibited by 5-fluorouracil. Now, the regulatory step in this pathway is CPS2, which gets activated by ATP and PRPP and inhibited by UTP and CTP, whereas OMPD carboxylase is inhibited by UMP and CMP. And also, dihydroorotate dehydrogenase is a mitochondrial enzyme. Rest other enzyme is cytosolic, and it is at the mitochondria wherein the ring closure takes place. Now, in contrast to purine synthesis, in which the ribose 5-phosphate is the primary substrate and purine synthesis takes place in the backbone of ribose 5-phosphate, in the pyrimidine synthesis, the ribose 5-phosphate is eventually added after the ring closure has occurred. Now, we'll look into the molecular arrangement that happens during the synthetic pathway. So the first step, as we discussed, that carbon dioxide plus glutamine in the presence of CPS2 leads to formation of carbamoyl phosphate, releasing glutamate molecule, utilizing one molecule of phosphorus. And this, in the presence of aspartate transcarbamylase, utilizes one molecule of aspartate, leading to formation of carbamoyl aspartate, leading to the formation of cut everything, restart from now. Bicarbonate plus glutamine forms CPS. This CPS in the presence of aspartate transcarbamylase forms carbamoyl aspartate. There's condensation that takes place in this step. This carbamoyl aspartate gets converted to dihydroorotate, wherein the ring closure takes place in the presence of enzyme dihydroorotase. This dihydroorotate is acted by enzyme dihydroorotate dehydrogenase, which is a mitochondrial enzyme, leading to formation of orotate. Now, in this orotate molecule, there is addition of PRPP, that is ribose 5-phosphate, leading to formation of orotidine monophosphate, and hence via enzyme OMPD carboxylase releasing one molecule of carbon dioxide, leading to formation of uridine monophosphate. Okay, so this was about the molecular changes that takes place during pyrimidine synthesis. Now, the deficiency disorder or the defect in the enzymes which are involved in the pyrimidine synthesis are as follows. Defect in the enzyme orotate phosphoribosyl transferase and OMP decarboxylase leads to orotic aciduria. Now, it is a hereditary orotic aciduria is a very rare disorder. In this, there is excess of orotic acid in the urine. The patient will have megaloblastic anemia and growth retardation. As I said, that any developmental delay means there is inherited metabolic disorder or enzyme deficiency. Now, the treatment of hereditary orotic aciduria is uridine-cytidine administration. So, this UTP is going to inhibit carbamoyl phosphate synthase 2 enzyme, hence decreasing the production of orotic acid in the body. The other syndrome, because of which there is increase in the orotic acid, is Ray's syndrome, and it is also is Ray's syndrome, and it is also called as secondary orotic aciduria. It happens because of the defect in the ornithine transcarbamylase of urea cycle. This enzyme is present in the urea cycle, and because of the defect in this enzyme, there is increased accumulation of carbamyl phosphate which enters the cytoplasm, and hence this excess carbamyl phosphate is utilized in orotic acid synthesis and hence increased in the excretion of the orotic acid. Now the salvage pathway of pyrimidine synthesis. As I said earlier, pyrimidine salvage pathway is more energy saving process, and the pyrimidine which is consumed via diet or during the recycle process of pyrimidine, cut it. Now we'll talk about the pyrimidine salvage pathway, in which uridine and ATP combines in the presence of uridine cytidine kinase enzyme to form UMP and ADP. Thymidine plus ATP in the presence of enzyme thymidine kinase from deoxy-TMP plus ADP. Deoxycytidine plus ATP in the presence of deoxycytidine kinase leads to formation of DCMP and ADP. This is the salvage pathway of pyrimidine. Now we'll talk about the catabolic pathway of pyrimidine, in which cytosine gets converted to uracil in the presence of enzyme cytosine deaminase, leading to formation of dihydrouracil in the presence of enzyme dihydropyrimidine dehydrogenase. In the presence of water and enzyme hydratase, it leads to formation of beta uridopropionate, which leads to formation of carbon dioxide and ammonia. Ammonia further enters the urea cycle, and thymine gets converted to dihydrothymine in the presence of the same dihydropyrimidine dehydrogenase enzyme. In the presence of water, again leads to formation of beta uridoisobutyrate, leading to formation of carbon dioxide and ammonia in the presence of hydrolase enzyme, and this ammonia enters the urea cycle. So the end product of pyrimidine synthesis is ammonia, leading to formation of urea, whereas purine, the end product, is uric acid. Now we'll look at the clinical case. A 4-year-old girl was brought to the hospital with complaint of weakness, failure of thrive, and delayed milestone. On examination, she was pale. So on laboratory investigation, hemoglobin was 6, which is less than normal, and megaloblastic anemia was detected. The other causes of megaloblastic anemia is vitamin B12 and folate deficiency, which were normal, and pyridoxine. As there was no response to the same, urine examination showed crystalline sediment of orotic acid. So as we have discussed that megaloblastic anemia along with the orotic acid in the urine is the classic feature of hereditary orotic aciduria. Okay? Thank you. हो गया